Hello everyone. In this class we'll try to learn about how bicarbonates and potassium they are handled by kidney. Reabsorption of bicarbonate. Reabsorption of bicarbonate occurs through the nephron except in the descending limb of LH. In PCT about 90% of the filtered bicarbonate is absorbed into the PCT. H plus secretion across the apical membrane of all of the cells occurs by both sodium potassium antiporter and hydrogen ATPase. The sodium hydrogen antiporter is the predominant pathway for H plus secretion, and the uses the lumen to the cell, that is sodium gradient, to drive this process. Within the cell, the hydrogen and bicarbonate are produced in reaction catalyzed by carbonic anhydrase. The hydrogen is secreted into the tubular fluid, whereas bicarbonate exits the cell across the basolateral membrane and returns to the peritubular blood. The movement of bicarbonate out of the cell across the basolateral membrane is coupled to other ions. The majority of the bicarbonate exit via symporter that couples with the efflux of one sodium with one bicarbonate, that is sodium bicarbonate cotransporter. In addition, some bicarbonate may exit in exchange for chloride. Carbonic anhydrase is also present in the brush border of proximal tubule cells. This enzyme catalyzes the dehydration of bicarbonic acid in the luminal fluid and thereby facilitates reabsorption of bicarbonate ions. So this is how the reabsorption of the bicarbonates happens. So there will be active secretion of H plus ions is going to occur through the active sodium hydrogen pump, and this hydrogen H plus ion binds with a filtered bicarbonate to form bicarbonic acid, which forms water and carbon dioxide, and that is reabsorbed. And when it is reabsorbed, again in the cell it forms H plus ions and bicarbonate ions, and bicarbonates are uh, transported to the capillaries, and that too it is occurs with the antipode with the chloride ions. In loop of Henle, the cellular mechanism responsible for reabsorption of bicarbonate in the thick segment of loop of Henle is very similar to that of proximal convoluted tubule. Potassium bicarbonate symporter in the basolateral membrane may also contribute to the exit of HCO3 from the cell here. In DCT and CT, intercalated cell secretes H plus ions and reabsorbs bicarbonate. Within this cell, the hydrogen and bicarbonate ions are produced by hydration of carbon dioxide, and this reaction is catalyzed by carbonic anhydrase. The H plus secreted into the tubular fluid via two mechanisms here. The first involves an apical membrane hydrogen ATPase, and the second, which couples the secretion of H plus with the reabsorption of potassium via hydrogen potassium ATPase, similar to that of found in the stomach. The HCO3, the bicarbonate, exists across the basolateral membrane in exchange for chloride, that is via chloride bicarbonate antiporter, and enters the peritubular capillary blood. This is how the bicarbonate reabsorption occurs in the late DCT. So one thing is, it is hydrogen potassium ion where it secretes H plus ions, and one potassium comes inside the cell, as a antiporter. The similar pumps, they are present in the HC gastric mucosa as well, and it can be secreted by active transport of H plus ions, H pump. So in a similar way, H plus ion is going to bind to bicarbonate, form carbonic acid, then water and carbon dioxide, which is reabsorbed, and in the cell again it gets converted back into H plus and HCO3, and HCO3 is antiported with the chloride ions. Coming to potassium handling, The kidneys play a major role in the maintaining of potassium balance. It helps in maintaining a balance between urinary excretion and dietary intake of potassium, thereby ensuring potassium homeostasis. Glomerular filtration. Because of potassium is not bound to plasma proteins, it is freely filtered by glomerulus. The potassium is the plasma electrolyte that is both reabsorbed and secreted into renal tubules. Reabsorption of potassium by proximal tubule. About 67% of the filtered potassium is reabsorbed by active transport in PCT. In PCT, the amount of potassium reabsorbed is a constant fraction of the amount filtered. Reabsorption of potassium by loop of Henle. About 20% of the filtered potassium is reabsorbed through thick ascending limb of loop of Henle along with the sodium reabsorption. Reabsorption and secretion of potassium by DCT and collecting duct. The distal tubule and the collecting tubule are able to reabsorb or secrete potassium. The rate of potassium reabsorption or secretion by DCT and CT depends on variety of hormones and factors. Most of the potassium produced in the urine is due to secretion of potassium in the DCT, which contributes approximately 75% of the excreted potassium. The potassium secretion from the blood into the tubule lumen is a two-step process which involves an active as well as passive process. The first step is uptake of potassium from blood across the basolateral membrane into the DCT by sodium potassium ATPase, and this is a critical step and is associated with active sodium efflux via sodium potassium ATPase. Then the elevated intracellular potassium is so achieved favors the net potassium diffusion at the apical membrane down the concentration gradient into the tubular lumen. Factors which are affecting potassium transport. The plasma potassium is an important determinant of potassium secretion by DCT and CT. Hyperkalemia, for example, which are resulting from high potassium diet or from rhabdomyolysis, stimulates secretion of potassium. Tubular secretion of potassium is markedly decreased in potassium and sodium depletion. In DCT, the potassium and hydrogen are both exchanged for sodium, in which reabsorbed, uh, reabsorbed, and the two ions compete for the exchange mechanism. Therefore, potassium excretion is decreased when H plus ion secretion is increased. Flow of tubular fluid. The rate of potassium secretion is proportionate to the rate of flow of tubular fluid through the distal portion of the nephron, because with a rapid flow there is a less opportunity for the tubular potassium concentration to rise to value that stops further secretion. Then mineralocorticoids, for example aldosterone, increases the potassium secretion by increasing the activity of basolateral sodium potassium ATPase, which increases potassium entry into the DCT, and second by increasing the permeability of the luminal membrane for potassium. Thank you.